Hi everyone, so today I'm going to talk about Discus and specifically their family tree, their genetics, maybe their phylogenetics, just about really what they are. So Discus are in the genus, is the genus Symphysodon, and they are cichlids. A lot of people don't recognise that. Their body shapes are quite different, but their closest relatives are Araru, Severums, which in the genus Heros. Both actually, those ones are called poor man's discus to some people, and that's quite interesting. They're quite closely related, whereas with other species you get poor man's something and then they're totally unrelated, like poor man's neon tetra, was it, or cardinal tetra, and that is the white cloud mountain minnow. So there are there's 3 recognised described species of Symphysodon. So the first one is probably the most well known. There's a lot of variants of it, or it's the one that contributes maybe more to so Symphysodon aequifasciatus is often called, well, I've seen it commonly called the blue discus, but I don't really want to use common names too much here because it can be a little bit confusing. So aequifasciatus, yes, is the sort of common species, will be associated with a lot of the domestic variants, but other species will contribute too, and to different extents would assume on how you actually sort of conclude the genus Symphysodon. So if you look at aequifasciatus, it's quite a colourful fish. It doesn't have any it has the numbers of barring, it has barring, usually, not all variants, well, captive bred variants you could say might not have, but it has this barring, especially when stress, and these are like stress bars. The next species, which is the really vastly different species, and this is Symphysodon discus. Symphysodon discus is called the Heckels, which is really confusing because you think discus, and then Heckel you'd assume, but it's just for some reason the common names don't work, and these aren't given the colour as far as I know for the common name, just the Heckels. So Symphysodon discus has that very distinct bar or barring that holds a lot better. I'm describing that so bad. Compared to aequifasciatus, it's very distinct, although it's a lot more it's a lot more distinct from aequifasciatus, but is a lot more closely related. The next species recognised is Symphysodon tarzoo. Now this looks very similar to Symphysodon aequifasciatus, but Symphysodon tarzoo differs from all the currently described species, as on the anal fin, which is this fin down here, it's probably a bit difficult to see. This is the so that's the ventral pelvic fins. That's below the side flappy fins, which are the pectorals. That's the ventral or pelvic fins. It's the same thing, I think, as far as I know. And this is the anal fin, which is this long one that goes along the bottom. In Symphysodon tarzoo, it has spotting, red spotting around that fin. That's the sort of defining morphological feature, and that is a very distinct species. It's very distant, you could say. It's more distant from Heckels and aequifasciatus, which are more closely related, but they show a lot more variance. And this is, it could be a number of things. Maybe it is convergent evolution, but maybe it's more logical, more parsimonious for Heckels, the Symphysodon discus, to be distant, to be sort of evolved their colour, their morphology, almost independently, because none of the others really have it. Although, so that's the main described species. And Symphysodon tarzoo, as much as I researched, and this was like a year ago research that I looked up, that they are smaller. They are a smaller, slightly smaller species, but it doesn't mean they stay miniature. They're not miniature. Discus are still big fish, especially domestics. They could get gigantic, like dinner plate size. So the next two species are undescribed. So this means they do not have a scientific name. They're obviously Discus, so then the genus Symphysodon, but they've not been described. So the first one, which is the main one, from my understanding, is Symphysodon sp. zingu. So this is... So just in editing right now, I just want to say thank you to Chris Engelsau, Sea Fish Essentials and Freshwater Life Project, just because he owns a lot of, quite a few of these brilliant photos, especially of the species I couldn't get, photos of. So I just want to say thank you for being able to use them. From the Rio Zingu, actually I have a lot of Rio Zingu fishes. The Zingu is probably one of my favourite rivers because it has so much diversity of localities, which is your plex. So the Rio Zingu variant is a distinct, possibly new species. From other studies, it looks like it's difficult to say how genetically different it is from the others. But it's quite unique in a way. It reminds me, just looking at it, it reminds me a lot of Symphysodon tarzoo in a way, but they're at opposite sides of the whole continent almost. Symphysodon tarzoo is more Peruvian side. So Rio Zingu is more towards sort of the mouth of the Amazon, but not actual mouth of the Amazon. It's very high temperatures, clear water. A lot of Discus don't come from such clear waters. As far as I know, especially I think the Heckels, tarzoo might seems to, I'm not entirely sure, I researched a while ago, but it's possibly considered that the adaptation to clear waters developed that sort of phenotype. So phenotype is how a species expresses its genes, and this is sort of the coloration, the body shape, stuff like that. The next undescribed species, and I don't find any references to it other than in papers, and it's probably sort of maybe misunderstanding or something. And this is Symphysodon sp. blue phenotype, and I've only found it in one paper, but it's quite an interesting concept if it actually exists. Okay, so this is the first paper that I'm going to look at, and this is looking at the mitochondrial DNA. So this DNA is inherited from the female side. Mitochondria are, I think, female side in most animals, at least I know it's that way in mammals. So this is sort of the powerhouse of the cell. It's a little part inside the cell, just to put it very simply. So the phylogeographic, so this is like the family tree in a geographic sense. The phylogeographic variant in the Amazonian genus Symphysodon, Discus. Teleostei is actually bony fish. So this paper, I'm not going to sort of read it out, but it generally recognises, as I scroll down, and then we go through results, and this is the family tree of Discus and sort of other related species. You can see very closely related is Uraru, and also Severums. Pterophyllum, that's your angelfishes. I've got a close-up of it now. Oh dear, I'll keep clicking in the actual thing. And I'm not too familiar actually. Hoplacus, that's the true parrot cichlid. So Hoplacus. So if we look at this, so this paper really looks at Symphysodon tarzoo, and there'll be other papers that really, I think, actually confirm this conclusion. So this is Symphysodon tarzoo, and it's not a very well-known species, but you can see it is distinct. It's found, the proven Rio Nanay ones are quite well known, but you can see this is Symphysodon tarzoo. It's in a very distinct lineage from Symphysodon aequifasciatus and Symphysodon discus. Interestingly, this paper mentions how Symphysodon tarzoo looks very similar to Symphysodon aequifasciatus, and but Symphysodon discus looks very different from the other two. Yeah, if we look at this placement, so this is a family tree, and you can see how closely placed they are. Of course, actually if I click on it, so now it doesn't work. Just going to see how close, doesn't really, it's a bit difficult to see if that's to a slightly different branch or not. Oh well. But as you can see, it recognises 3 species in the genus Symphysodon. So that's 3 different discus species. Let's move on to another paper. So let's go on to actually this one. So this one is this is published in, I don't know how reliable this paper is. And I should mention the previous paper, I believe they got the other sequences from the MCI database, which is a bit hit and miss. So if I scroll up, this was published in this journal I've never heard of. And there's genetic variation in domesticated discus. And this is actually the only paper I could find on the topic. So I don't know how, I would read really in detail, but it does look that they have actually extracted the DNA themselves. So it does rely on them using reliable identifications. I don't know if there's any actual... So here, so if I go up, they use the common names, and it kind of bugs me, but then you can't really do much with when you've got varieties and you have to keep listing the scientific name, and you've got so many. And with discus, so this really does look at the captive bred variants. So if we go here, so this is your popular captive breds, and these are all recognised more or less largely as Symphysodon aequifasciatus. And you can see it really places here. And one thing I should mention with phylogenetics is you can flip. So you can flip these branches. So where 85 is down there, 96 is up there. So it's really difficult to explain it. So sometimes you have to look at it as, so this doesn't mean that this, although these are different lengths, so this would infer that this is more closely related to this side, I think. I don't really like using lengths on phylogenetic trees, but then I only do so many. So this is aequifasciatus, and here's your little Heckels. And then we've got browns. So the brown would be haraldi, Symphysodon haraldi. So you don't really see the wild of that species in stores. But they do appear, and sometimes they're either a species in papers, they're synonymized with either tarzoo, which I don't think is correct, or with aequifasciatus. You can see it's actually it's more closely related to Heckels. I think other papers from memory actually show that. And the blues is aequifasciatus. So that's a bit odd. To me, that looks like Heckels are nested within. But then if I bring up that previous paper, so this is the Symphysodon tarzoo paper, as I call it. This does show that Discus is more or less, seems to be possibly nested within aequifasciatus, maybe. So this is something called, Symphysodon aequifasciatus is polyphyletic, and that's not really ideal. although this could be, there is something called SP blue, and that could actually be that. And that would, if we look at the variants, not all of these variants, it can be a little bit complex. So look at pigeon blood. So pigeon blood is not a different species, but what this paper actually mentions somewhere around here is that pigeon bloods, where did I find it? Pigeon bloods, it could be due to levels of hybridization. So because this is based on DNA and similarities of DNA, where it's hybridized, it's going to have a mixture of DNA from either parent, and that's going to shove it out here. So it doesn't mean that this is a different species. So this, there's a lot of interpretation to this, but it does show that the greens, which is Symphysodon tarzoo, are independent from the others, and that's got very high support. So this support base, these numbers show how highly supported each branch is in its placement. 99 is really high. Ideally we want above 70, but you can't always get it, and it depends how you're actually calculating support. There is mathematical stuff basically. So before I bore people out of this paper, which I think is quite interesting, this suggests that that's the haraldi. Rose reds could either be maybe a hybrid of haraldi, maybe haraldi, and blues could be aequifasciatus, or it could be that unknown blue species which is described in this paper. So if I get rid of that one, because I'll leave it up for a little bit, I might delete it if I don't. So this one actually recognises several different species. So this is the abstract, and this is a review. So this is Molecular Perspective on Systematics, Taxonomy and Classification of Amazonian Fishes of the genus Symphysodon. So this was published in a journal that they've given only the initials for, which is, I'm going to assume, what did it say? No. So it just says evolutionary biology, so that's all I can assume of it. So this one looks at, okay, so this is actually more than a review. So it says here, the results indicate Symphysodon is composed of 4 genetic groups. So that is group purple, phenotype Heckels and Abexi, group green, phenotype green, group red, blue and browns, group pink, populations of Zingo and Cometa. So was it So we therefore conclude that Symphysodon comprises of 5 evolutionary significant units. Symphysodon Discus, so that's the Heckels and Abexi. Symphysodon Aequifasciatus, which is your browns, which is a strange one. Usually brown is associated with haraldi. Symphysodon tarzoo, which is your greens. Symphysodon SP1, which is your blue phenotype, and Symphysodon SP2, which is the Zingu one. So the Zingu one's actually quite interesting. Let's see if I don't remember this having a phylogenetic tree. But then the previous paper, so I have gone back to this one, because there's this chart here which shows how distinct the greens are from the others. So this is really high difference opposed to these ones, opposed to the other species with each other. So the greens are really distinct. And this shows variance in the species. It's quite interesting actually. Heckels, so these Heckels have very little genetic diversity, it seems, because they used the worst calculation anyone has to ever be put through, which is Hardy-Weinberg. I still struggle with it. It has very little genetic diversity compared to, and also the greens they're moderate. Zingu next to not much at all here. So this is at species level, this is at population level. I thought it was quite interesting anyway. So maybe it's not looking at phylogenetics, but I think, because I like trees, producing a tree makes it very interesting. Maybe I should have read this more. This shows actually water parameters, I think. So, what's that P probability? Oh well. Well, pH ain't going to be that high, that low, so I don't think. Diagnosis of phenotype. Okay. But it's just interesting how these actually conclude using different methods. pH and conductivity. Table based on Bleher. Okay, that's quite useful actually if people I'll be linking these below, but if people want to look at parameters, maybe that might be of use. There's also other papers you can find parameters. I have a Excel sheet on it. So this is the species that they actually conclude. Symphysodon Discus, which is Heckel's, and this includes synonym, so this means that species was shoved in, well, it's no longer considered a species and is placed in Symphysodon Discus. We generally don't recognise subspecies, so lob that off a little bit. So that's Willa Schwartzii, is that how you'd say it? So they recognise Symphysodon Discus, Symphysodon tarzoo. So there is also this synonym, so one that's been shoved in, Symphysodon Discus aequifasciatus. So this is one of the the paper actually goes more into detail about this, and I read it very late, about 1 a.m. on a Saturday night. That's how boring I am. So this is a particular, it's not aequifasciatus itself that is recognised as a species. This is a particular group of specimens, I'd say. Symphysodon Discus tarzoo, Symphysodon aequifasciatus. So this is another subspecies of Symphysodon aequifasciatus. Symphysodon aequifasciatus haraldi. And this goes, so it says here, so these were specimens collected and they were given this locality, although this locality doesn't seem to be correct. So the assumption is the Symphysodon aequifasciatus haraldi collected in this area are actually Symphysodon tarzoo. And Symphysodon aequifasciatus mentioned in Bleher's paper, and I'm butchering pronunciations here. So then we've got Symphysodon aequifasciatus, that is recognised, and I find it brown just creates confusion, but then I should have explained the blue and that lot is common name, so brown phenotype doesn't mean it's now the brown discus. So this now includes the Symphysodon Discus variant. Barn isn't used that much. I don't think it's actually recognised in the International Commission for Zoological Nomenclature, so how you're allowed to name things. So it's interesting they actually used bar. It's just weird rules. So also Symphysodon aequifasciatus axeroides. And this is if the actual type locality of those fishes collected that species is named after is found in the Rio Tapajos. And Symphysodon haraldi, that is mentioned in Bleher. You need to write I think it's easier somewhat to sort of write it down, maybe make a chart, because there's a lot of names that practically almost the same being shoved in it, and I think it's just due to localities or maybe very similar looking fish, because a lot of them do look very similar. So Symphysodon SP1, which is the blue phenotype. This is not these two are not described species. So this is Symphysodon haraldi in Bleher, and also Symphysodon aequifasciatus in this paper. So it suggests that maybe misidentification. You've also got Symphysodon SP2, which is the Rio Nanay species, and I don't think there's much discussion on whether there doesn't seem to be much research on them, and I guess it's a very niche area, but it's a very highly fished area, the Rio Nanay, but it might be from a more obscure area. So I might just quickly look at biological groups, genetic difference. Genetic diversity, okay. So it doesn't look at phylogenetics, but it's still interesting. And why, I don't know entirely why what's it called? The Xingu one really isn't studied much. Because I think it could be a new species, but it's a bit difficult to say if you get what I mean. And this is just based on the papers I'm reading and I've read. So there's the last paper. I'm not going to go too into detail with it. It doesn't recognise tarzoo, Symphysodon tarzoo. So it recognises here. This paper only recognises Symphysodon haraldi, Symphysodon Discus, and Symphysodon aequifasciatus, it seems. Nice. Here. So this shows the different, I believe the different, so this colours correspond to phenotypes. Now I really like this actually, it's quite useful. Oh well. So this shows as you go through this sort of the Amazon, you get different variants, and so all the papers have concluded that, well, not all the papers, one I've read and two I think conclude that Discus are going through speciation, so they're not that distinct as species. You could say they are subspecies. Maybe Symphysodon tarzoo is completely distinct. I don't think. The Xingus might be, but as you can see, so this is the Xingo variants, and as you go through, these are all quite similar. I think, because I'm not sure about how similar those are, but Symphysodon tarzoo is very distinct at the end, and it kind of reminds me a little bit. Firstly, I think there's a barrier likely there that I'm not sure how sympatrically they're found, because a lot of people say that there's hybrids of tarzoo, but there's no papers almost to suggest that there is in the wild, and it's just that there's a barrier, and a lot of these aren't being described properly, identified properly. And this looks like a ring species, almost. Or this is just real, so what's the word? Allopatric speciation. So this is where a species evolves, where location or the environment is the barrier preventing the mixing. So these will probably be, they can hybridise, but they might not be able to, and actually explains, but these are more distinct, actually explains, I found the tarzoo, a lot more, they weren't very tolerant of Heckels, and I wonder whether that distance means they don't recognise each other, because you get hybrids of Heckels with the others, and you probably do even of the tarzoo, but they weren't as they were ill-tolerant of the Heckels. It might be that they're so distinct they don't actually understand each other as you'd expect. So I think that's quite an interesting thought anyway, because I find different variants of Discus they probably do not so sometimes it's worth treating different populations as different species. They might not be able to understand each other, and I think it's particularly clear in ones that don't bar and ones that do. Whether the ones that bar just think what's wrong with the ones that don't. They don't seem to be giving these behavioural cues, and they're very intelligent fish. They're quite they're really smart. They can recognise faces, so I think it's quite interesting anyway. And while I'm not intensely literate on the topic of discus, there's not too many papers, and I just think it's quite interesting, and I study catfishes, so it's a bit of a different one. So it's quite complicated, I guess, the actual phylogenetics of species, and maybe it is because of how they are speciating and how similar many of them actually do look to each other. Obviously, when you've got the domestics, those solid pigeon bloods and the pigeon blood relatives, they possibly did occur from hybridization, which might cause the results like that. And it might, if you were doing say morphological studies, maybe with discus there's a little bit more sort of misleading assumptions, and a lot of historical phylogenetics would be done using morphology. So it is just an interesting topic in general, and while I think the domestics are really popular, I think the wilds are just as stunning and maybe underrated, and that's why maybe there isn't so many studies, because there's loads on the actual there's loads of things on the care, the breeding. There's studies on the aquaculture, the ecology, and the ecology is probably another topic I actually need to go into, because it's a lot more different than people think. The intelligence, and the actual feeding in captivity and in the wild. So it's quite interesting. Obviously there's species that we're not quite sure where to place entirely, like haraldi. A lot of people aren't entirely sure about that species, whether it exists, whether it doesn't. Tarzoo seems debatable, and I believe that's due to sort of the nomenclature that was originally used for tarzoo, because although I showed that paper, the actual description of tarzoo was a lot longer ago. Sometimes those old papers can be a bit funny. A lot of the actual descriptions can be a little bit of a strange one. Locality is obviously important. Locality can be a little bit troublesome. If a type has a lot of species are described using a type, and if the type isn't the same, if the type isn't sort of from the right locality, it can cause a lot of issues. And while I'm more experienced with localities, discus are they are interesting, but I'm not so familiar on their genetics, and especially when it comes to cichlids in general. I don't study cichlids, but I can apply some of my knowledge from localities, and I think cichlids, I find their changes a little bit more subtle, but then the more you spend with a species, the more you understand them and their morphology, and that's why you see people even people that judge the fishes, they see a lot more differences that you might not see unless you spend a lot of time with them, because they notice the minute details. So I'm probably going to finish this video here, and maybe the next few I will talk about diets, because I think that's a little bit more interesting, and I've got a little bit good experience with looking at diets from different species, although generally I study a lot of evolution. So yeah, and thank you for watching. If you like my videos, then comment, subscribe, whatever, and I will do a video eventually when I reach 1,000, which hopefully won't be too long, and I don't know what people would want for it entirely, because I don't entirely want to, I don't think I could do a live stream for a while. It takes a lot of energy. I've done one before and it was 2 hours and I was absolutely exhausted afterwards. But I could always, I might do one introducing myself, because I've never really entirely done that. And also I need to do a tour of my collection, my wet specimens collection. There's a lot, and there's more I need to preserve. There's a lot more I need to preserve because they're in the freezer. I need to find a jar for an adult barla shark. There's a photo of him and he's about that big. Problem is the largest jar I've got is this big, and I don't want to really fold him in and break a load of bones. So that will be really fun. And then I've got some weird things in the freezer that, like, I've got some rats that I thought, oh, Quet will be able to eat that. She's big enough. No, she isn't. There's no chance, I think, in any matter. Like, they're like medium rats. There's no way she's going to be able to eat them, ever. She's a Western hognose, so she's quite small. So I'll probably end up preserving them, because I think learning to actually properly do taxidermy might not be, like, in the first full flat it's going to get quite smelly, and also there's a lot of hazardous toxic chemicals involved, but I can always preserve in a jar, and that's kind of easier. Although there is toxic hazardous chemicals, it's a lot easier. And I do need to organise this. I want nicer, stronger shelving. I do worry there's a lot of weight on each of the shelves. None of them seem to be bowing now. Yeah, they're right next to me. But anyway, thank you for watching, and goodbye.